Hey guys, so we've gone through basic patterns of inheritance. Now we're going to go through a little bit more complex patterns of inheritance in this video today. So, remember we went over first our Mendelian principles. Mendelian principles are generally ruled by a single gene, and we're going to have two alleles that we're talking about for the most part. Today we're going to talk about instances where there might be multiple alleles and how that works with inheritance. So keep in mind, the relationship between a gene and a trait is not necessarily simple. In fact, most of the traits that you recognize in your body are controlled by multiple genes, and there may be multiple reasons for why these are interacting with each other or presenting in the phenotypes as they do. So, three important types we're going to start with. Incomplete dominance, that's when we have a blending of traits, and we'll notice our pink flower here is an example of incomplete dominance. Our codominance, this is a hybrid with both traits. Frequently we'll talk about cows and their spots as an example of codominance. And multiple alleles, more than two allelic forms. So, blood types is a great example of when something has multiple alleles. It can also be an example for codominance too, but we will talk about that. All right, so incomplete dominance. Again, we notice we have the dark pink, we have the white, and then the offspring of this particular cross would provide us with a blending of these two traits. I want you to note that when we do write out incomplete dominance, you may see the notation for these types of alleles a little differently depending on the source or the textbook or the question that's being asked. So sometimes we will see big R and little r as in our traditional forms of regular laws of dominance. Sometimes we'll see two capital letters, and sometimes we may see notation as like an R1 and R2. So just be careful and keep an eye out for that when you're doing problems with incomplete dominance. Now in codominance, we are seeing the expression of both alleles at the same time in the phenotype. So for a very common example of this is roan cattle, where we have potentially a white and a tan cow crossing, and they're going to produce cattle with both tan and white spots. So we see in this particular form, we are writing with two capital letters, and that's pretty much always the case for codominance. We will see two different capital letters presenting themselves in our genotypes. All right, multiple alleles. An individual may only carry two alleles for a given gene, so you only have two copies of every gene, but in a population there may be any number of alleles for that gene. So for example, the ABO blood type gene is an example of a gene with multiple alleles. So our three alleles that we can have are big IA, big IB, or little I, and if you have whatever combination of that will determine your blood type. So someone who is type A blood will have big IA, or big IA, little I. And that little I is going to be representative of the type O trait. And in order to have the type O trait, you need two copies of that because it is recessive. Now I have a whole video on blood types if you want to get into this a little further and how those are inherited. So be sure to check that out if you would like. Now let's talk about interactions of different genes. So pleiotropy is when one gene can affect more than one trait. Epistasis, we have two separate genes controlling one trait, but one gene masks the expression of the other. And polygenic inheritance is when characteristics resulting from the blending of separate genes result in this continuum or a blend. So like skin color is an example of that. So again, short way to remember these. Pleiotropy, one gene, many effects. Polygenic inheritance, many genes determining phenotype, and an additive effect. So we can have like a continuum, a scale. And epistasis, one gene controlling the expression of another gene. So some examples of each of these. Pleiotropy, cystic fibrosis. We know generally this is a mutation of just one gene, but we have multiple phenotypic effects in the organism. So you may have problems absorbing things in your digestive system, but you also have this mucus buildup in your lungs, and so you have problems with your respiratory system, all caused from one gene mishap. The agouti coat color in mice is going to be an example of epistasis, and we'll talk more about this one in class and how these genes are interacting with each other, because you're going to have the albino color, the black color, and then if you have certain genes being working together, you will get the tan agouti color of this mouse. Now, polygenic inheritance, there's a lot of examples of these. Height in humans is a big one. So we have a scale. We don't have like one gene that determines whether you are one version of tall or one version of short. And you know that two tall parents can produce an even taller child. So that works through polygenic inheritance. Now, I also want to talk about the multifactorial basis of disease. So alcoholism and heart disease are two diseases that you can inherit genes for or propensity for, but you won't necessarily become an alcoholic because you have those genes. So I wanted to pull up something from the NIH really here. So this is the genetics of alcohol use disorder. It's a little article in the NIH. You can look it up yourself. And I wanted to read this particular part right here. Research shows that genes are responsible for about half the risk for alcohol use disorder. So therefore, Genes alone do not determine whether someone will become an alcoholic. Environmental factors, as well as gene and environment interactions, can account for the remainder of the risk. So that's 50% is determined by your genes, but the other 50% of whether or not you're going to become an alcoholic is determined by outside factors. So that's what our multifactorial basis of disease is all about. And there's many diseases that relate to this. All right, so as we're loading back up, let's talk about penetrance. This is the proportion or percentage of a population of individuals in a group with a given genotype that show the expected phenotype. Now, just because you have a genotype may not mean you will always show it, and that could be due to environmental factors, as we just talked about, or due to other reasons that we don't know about yet. So, for example, patients with the mutant BRCA1 gene, some of them will develop breast cancer, not all of them, and we don't know exactly what's causing some of them to develop breast cancer and others not to, but the penetrance of that is something we can measure. So those who do have the BRCA1 mutant gene developing breast cancer, we can calculate the penetrance of that. All right, so let's talk briefly about linked genes. Now remember, unlinked genes are genes that are located on different chromosomes or so far away on one chromosome that I have a really high recombination frequency, so they won't be inherited together. A linkage group is a set of genes at different, battery's running low. A linkage group is a set of different genes, genes at different loci on the same gene, that are inherited together for the most part, except for crossing over events. So if there's a linkage group, we're going to see different patterns of inheritance than we would in a traditional unlinked gene sense. So our Mendelian patterns of inheritance are assuming that these genes are assorting independently, there's segregation, and these genes are unlinked. So, really quickly, sex linkage. Now, when we talk about sex linkage here, we're mostly referring to genes that travel on the X chromosome, which is a sex chromosome. Now, colorblindness is a really popular example for this, and I do have another sex-linked traits video, which you can watch as well if you need to. So, a normal woman, for example, for colorblindness, would have this particular genotype. If she is a carrier, meaning she can carry the trait and pass it on to her offspring, but she does not display it, she would have this genotype. The little b is the trait, the mutant allele for colorblindness. In order for her to be colorblind, she needs 2 copies of that mutant allele, those 2 little b's. So it's rarer for women to have X-linked or sex-linked disorders, especially if the gene is recessive, which generally it is. Men, however, have a higher instance of these sex-linked traits showing up as actual disorders in them because they only need 1 copy of that gene in order to display the trait. Remember, the Y gene or the Y chromosome is not really important here. We're just looking at whether or not they have the gene or the unaffected gene on the X chromosome. So males who are normal will have just a 1 copy of the regular gene. Males who are colorblind will have just 1 copy of the mutant gene. Now, males cannot be carriers for sex or X-linked sex traits because in order to be a carrier, you're not displaying the trait yourself, but you could pass it on to your offspring. Men are always going to display the trait if they have it. All right, so remember sex-linked trait tips. Again, you can watch the sex-linked video if you want. Males cannot be sex-linked carriers. They're not carriers. They either have it or they don't. And sons do not inherit sex-linked traits from their father. Why? Because the males only are going to deliver the Y chromosome to their sons. Not literally deliver, but yeah, you got it. And it's very uncommon for females to display these sex-linked traits. It's more common for males. Okay. So, really quickly, let's talk about pedigrees. Now, pedigrees are useful tools for determining inheritance of traits. This is a really quick key to common notations or in pedigrees. A normal female will be just a plain circle. A normal male will just be a plain square. If you don't know the sex of the individual, or if it's a normal individual, it will be a diamond like that. If the female has a phenotype of interest, the disease, for example, from mapping like Huntington's disease in a family, she will be shaded in. Now, a key point in looking at pedigrees is sometimes if the female is heterozygous for the recessive allele, she will be half shaded. Now, this is not always the case. You can't always assume that the female is not a heterozygous carrier if she doesn't have a shade in it. So when we do pedigree problems, don't always count on this little half shade part. Now, a marriage or mating is represented, or a union is represented by just 1 line. Double line if there's mating between relatives, and then generally siblings are ordered right underneath the parental units. So we'll have our firstborn here, and then it will go in this direction, and they're related. You can see through this line here that goes up to the parents. These are monozygotic twins or identical twins, and these are digozygotic twins. But we'll take a look at pedigrees later on. Okay, so let's do a really quick practice question, and then we'll be done for our patterns of inheritance video. So consider a gene with 4 alleles. This is a multiple alleles question. The alleles are A1, A2, A3, and A4. In a cross between an A1, A2 parent and an A3, A4 parent, what is the probability that a particular offspring inherits either A1, A3, or both? So let's take a look. The best way to set this up is good old-fashioned Punnett square, and the one parent down here will have A1 and A2. The other parent at the top will have A3 and A4 that they could donate through their gametes. We'll fill out the Punnett square, and then as we look at it, we're like, oh, look, this one has either A1, A3, or both. It has both. This one has A3. This one has A1. So it's asking what is the probability that a particular offspring inherits either A1, A3, or both. And this is very easy. We can see that it's 75% or 3/4. There we go. Done. Practice question 2. A woman is affected with a trait due to a dominant mutant allele that shows 50% penetrance. So a penetrance question here. If she has a child, what is the probability that it will be affected? I think it's supposed to say that the child will be affected. What's that probability? So let's take a look. We can set this up by looking at the woman's genotype. So she's affected with a trait, dominant mutant allele. So we have the dominant mutant allele. I put it in red because we know that this is the thing that's giving us the trait. If we want to see the percentage of the child being affected, so we first have to figure out what percentage the child has of actually getting this allele. So we're going to ignore the other parent for now because we don't know what their genotype is. But if we're just looking at the mom, she could have a 50% chance of giving this dominant mutant allele to her children. Now we could fill in with a homozygous recessive or whatever you want to see the difference, but there's still going to be a 50% chance that her particular offspring will give the dominant mutant allele. But we have to remember that it's 50% penetrance. So when we're doing this, remember we multiply our probabilities by each other. So if there's 50% penetrance, we have to multiply our 50% chance of giving this to the child by the 50% penetrance. So 1/2 times 1/2 equals 1/4. There's a 1/4 chance, or 1 in 4 chance, or a 25% chance that the child will be affected. All right, so I have some other videos on pedigrees, sex-linked traits, blood groups, and of course, wizard inheritance, which if you're interested, you can watch. But other than that, we are done. See you later.